Eukaryotic cell cycle consists of several phases: interphase, M phase, and cytokinesis. Interphase is broken down into Gap1 phase, synthesis, and Gap2 phase. M phase, also known as mitosis, is broken down into prophase, prometaphase, metaphase, anaphase, and telophase. Interphase begins with Gap1 phase. This is where the majority of cell growth occurs. Some organelles duplicate and there is a protein buildup. mRNA synthesizes and DNA is found in the form of uncondensed chromatin. There's a checkpoint that occurs in this phase in order for the cell to commit itself to division. If the checkpoint is not passed, the cell enters a phase called Gap0 where all division is halted. Then there is synthesis. DNA synthesizes and is still uncondensed in the form of chromatin. In the next phase, Gap2, there is another checkpoint. This checkpoint occurs to check if DNA is synthesized without any errors. The cell then reaches its potential growth, the nuclear envelope encloses the nucleus, and protein synthesis occurs for the duplication of centrosomes. The duplication of centrosomes each pair with a centriole. Mitosis begins first with prophase. In prophase, the nucleoli disappear, and then meiotic spindles begin to form from each of the two centrosomes. Short microtubules called asters also extend from them, forming a star-like shape around the centrosomes. DNA condenses into sister chromatids, with each chromosome now having two sister chromatids connected in the center via a centromere. Next is prometaphase. The nuclear envelope begins to fragment. A protein structure called kinetochores now form in the centromere. Microtubules that connect with kinetochores become kinetochore microtubules, and they jerk the chromosomes back and forth like a tug-of-war to maintain balance. Non-kinetochore microtubules interact with both ends of the meiotic spindle. The next phase is metaphase. Chromosomes align themselves at the center, equidistant from both poles in the center region called the metaphase plate. A checkpoint is then performed to ensure that the chromosomes are aligned and connected correctly to perform division. Centrosomes are now at opposite ends of the meiotic spindle, forming poles. Kinetochores are attached to kinetochore microtubules coming from opposite poles. Next is anaphase. Cohesion proteins are cleaved, causing sister chromatids to split into two. Each chromatid becomes an individual, full-fledged chromosome as it is dragged by the kinetochore microtubules to the opposite ends of the cell. Cells elongate as non-kinetochore microtubules lengthen via tubulin polymerization. By the end of anaphase, the two regions have a same and full collection of chromosomes. The next phase is telophase. Two daughter nuclei form in the cell. A new nuclear envelope forms from fragments of the old one. A nucleolus reappears, and chromosomes again become less condensed. Spindle microtubules are depolymerized. Lastly, there is cytokinesis. Cytokinesis happens simultaneously at the end of telophase. Actin and myosin filaments form a ring along the plasma membrane. The filaments contract, thus constricting the cells into a cleavage furrow, which eventually splits the cell into two daughter cells. A similar process of cell division is meiosis. Unlike mitosis, meiosis consists of meiosis 1 and meiosis 2. Meiosis 1 is broken down into prophase 1, metaphase 1, anaphase 1, telophase 1, and cytokinesis 1, whereas meiosis 2 is broken down into prophase 2, metaphase 2, anaphase 2, telophase 2, and cytokinesis 2. Before we move on, here is some more information on interphase. During interphase, DNA replicates, forming a tetraploid cell. Centrosomes duplicate and each has two centrioles. In prophase 1 of meiosis 1, chromosomes condense, homologs loosely pair along their lengths, aligned gene by gene. Synapsis occurs. A synaptominal complex binds the homologs together, which makes tetrads. Tetrads are held together at the points where they cross, called chiasmata. Crossing over of non-sister chromatids occurs at a specific gene locus to increase genetic variation. The nuclear envelope breaks apart. Microtubules extend from the centrosomes and attach themselves to the kinetochore proteins on each chromosome centromere. Microtubules that do not attach to any kinetochore are non-kinetochore microtubules, which interact with each other from opposite poles of the cell. In metaphase 1, pairs of homologous chromosomes are arranged at the metaphase plate, with one chromosome of each pair facing each pole. Both chromatids of one homolog are attached to kinetochore microtubules from one pole. Random assortment then occurs, in which paternally inherited chromosomes don't have to go to the same poles. This increases the possibility of different placement in the cell. Next is anaphase 1. Proteins are broken down, which separates the homologs. But since sister chromatid cohesion persists at the centromere, the chromatids do not separate. Each homolog is then pulled by kinetochore microtubules to opposite poles. In telophase 1 and cytokinesis 1, microtubules depolymerize. Each pole now has a complete haploid set of duplicated chromosomes, each chromosome with 2 sister chromatids. In cytokinesis, myosin and actin microfilaments constrict, which causes the formation of a cleavage furrow, which creates 2 haploid daughter cells. Once these daughter cells are formed, new nuclear envelopes form, and no chromosome duplication occurs between meiosis 1 and meiosis 2. Once cytokinesis 1 is complete, the cell begins the meiosis 2 process. Meiosis 2 begins with prophase 2. During prophase 2, centrosomes duplicate, the meiotic spindle reforms, nuclear envelope breaks apart, and chromosomes move towards the metaphase 2 plate. In metaphase 2, chromosomes are aligned along the metaphase 2 plate. Because of crossing over in prophase 1, the 2 sister chromatids of each chromosome are not genetically identical. Kinetochores become attached to kinetochore microtubules, and the cell reaches another checkpoint. The checkpoint checks if the kinetochore microtubules have attached properly to the chromosomes. Next is anaphase 2. Each chromosome separates into individual sister chromatids due to the breakdown of cohesion proteins. Chromatids move towards opposite poles by the shortening of the kinetochore microtubules. When they reach opposite poles, they become individual chromosomes. Next is telophase 2 and cytokinesis 2, the final phases of the process. The nuclear envelope and nuclei reform and chromosomes become less condensed. Spindle microtubules are depolymerized. In cytokinesis, actin and myosin microfilaments constrict to create a cleavage furrow once again, which produces 4 daughter cells, each with a haploid set of unduplicated chromosomes. The 4 daughter cells are genetically distinct from each other and their parent cells. This completes the meiosis 2 process.